Assalamu alaikum everyone. In this video lecture, we will be discussing the electron micrographs of animal cells. So what are electron micrographs? These are the pictures taken from the electron microscope. So simply, we will be discussing the pictures of the animal cells taken from the electron microscope. So this is the picture of an animal cell taken from an electron microscope in front of us, and this in this picture, it's very difficult to identify different cell structures. So I will give you some tips and tricks to identify different cell structures when you see a picture of an animal cell that is taken from an electron microscope, or when you see an electron micrograph of animal cell. So guys, we can see that in the center of this cell, this large or huge structure is the nucleus, right? And nucleus has its membrane, so we can label this membrane as the nuclear envelope. As we all know that the membrane of the nucleus is the nuclear envelope. And we also know that there are pores in the nuclear envelope or holes in the nuclear envelope. So over here, this structure, over here we can see that there's a hole or pore in the nuclear envelope. This is the nuclear envelope, which is a double membrane, and it's coming like this. So there's a break or a pore in the nuclear membrane, which can be labeled as nuclear pore. So guys, you know that nuclear membrane is a double membrane. So basically this is a double membrane going on, right? Which is the nuclear envelope. So we have labeled the nuclear envelope and we have labeled the nuclear pore. Inside the nucleus, we can see the chromatin, and at some places, chromatin is darker like this. And at some places, chromatin appears lighter. So the dark chromatin is known as the heterochromatin, which is highly coiled chromatin. And the light chromatin, what do we call the light chromatin? It's known as the euchromatin. It's loosely coiled chromatin. As you all know that chromatin is made up of DNA coiled over histones. So wherever there is loose coiling of the DNA, euchromatin is formed, and wherever there is tight coiling of DNA, heterochromatin is formed. Right. What other structures can we see in this cell? So guys, this is the structure that we can see, and more such structures like this. So guys, these structures are mitochondria. How do you recognize mitochondria? Mitochondria will have a sausage shape or an oval shape or spherical shape, right? This is an oval shape. This is kind of spherical, and this is kind of long sausage like. So these structures are the mitochondria. How do we actually recognize the mitochondria? When we will see the mitochondria, we will see some dark regions and some light regions. And if we look very carefully, we can see the lines in the mitochondrion. These lines are the lines of the Christie, right? So you will see some light structures, some dark structures in the mitochondrion, and it will have a characteristic shape. It can be sausage-like, it can be spherical, or it can be oval shaped. So these are the mitochondria. This is how we recognize the mitochondria, right? And over here you can also see some dark lines in this structure and some areas which are white or lighter in the mitochondrion. Then you can see these sacs, these long sacs, and at the outer edge of these sacs we can see dark dots, right? So basically this is characteristic of rough endoplasmic reticulum. So guys, if you see long sacs, very long sacs, and you see dots on the external surface, black dots on the external surface of these sacs, this is definitely rough endoplasmic reticulum. So guys, we can label these as what? Rough endoplasmic reticulum, or RER. So how do we identify RER? These are the long sacs, and What else do you see? Black dots on the surface or on the external surface of these sacs or pouches, right? What else can we see in this cell? If we look very carefully, so we can see these curved pouches, right? We can see these curved pouches. These are Golgi apparatus. If you look at this carefully, these are the curved pouches which are going like this. These are kind of blur, but I'm highlighting them for you so that you can understand, right? And you can also see vesicles coming out from the Golgi apparatus, right? This is also a pouch or a sac, and you can see the vesicles coming out of the Golgi apparatus. So guys, this is what Golgi apparatus with Golgi vesicles. Golgi apparatus, and around the Golgi apparatus or the Golgi bodies, you can see Golgi vesicles, right? What else do we see in this cell? We can see the lysosome. So this large sac over here is the lysosome, which appears white over here. Guys, lysosomes can appear dark as well. Lysosome can appear lighter or darker under the electron microscope, right? So we can see mitochondria, we can see the nucleus, we can see the lysosome, the RER, right? Let's move on to the other electron micrographs. So guys, in this picture, I have already placed the labeled lines. So what do you think are these structures? These are mitochondria, right? And what do you think is this structure? This label is for the plasma membrane or the cell surface membrane. And if we look at this structure, this was not visible in the last last electron micrograph, right? What are these structures? Whenever you see two cylindrical structures at right angles, these are centrioles, right? So basically, this is a cylindrical structure, and you can see over here a spherical structure which will be going back like this. So basically, these cylindrical structures at right angles are called centrioles. These structures are found in animal cells close to the nucleus, right? So this label over here is for the nucleus. This structure is the nucleus, and this structure is the lysosome, right? So we are done with the labels. These are mitochondria, these are lysosomes, right? Lysosomes can also appear darker under the electron microscope. Okay, let's discuss some other electron micrographs of animal cells. So guys, this electron micrograph of an animal cell in front of you is somewhat different in a sense that it contains a lot of SER. So how do you recognize SER? These two electron micrographs both contain extensive SER. So guys, how do we recognize SER? SER will be pouches or sacs which will be shorter, and they will be dilated or widened. They will look like large vesicles which are irregular in shape. Normally vesicles are circular in shape, but SER looks like vesicles that are, we can say, flattened or irregular in shape, or we can say dilated. So this is how you recognize SER. SER will look like vesicles, but actually it's not. These are not vesicles, these are SER. Over here you can also see that these are SER. In this electron micrograph, the SER looks dark. So the students think that this is RER. Basically, over here, these are actually the RER. Why? Because I told you that RER will consist of flattened sacs and there will be black dots at the external surface. This is the feature you will use to recognize RER. You can see in the RER as well, the boundaries of the sacs are dark, but ribosomes appear separate. So ribosomes will always appear separate. You don't have to consider the dark boundary of the sac to identify that it is RER. To identify RER, you need to have a look on small dots at the external surface of the sac. So guys, in this electron micrograph, these are RER with ribosomes attached to their external surface, and these pouches which look widened, dilated, vesicle like, these are SER. So guys, this is how you identify SER on the electron micrographs of animal cell. Over here, these are SER, right? And you should know that SER or smooth endoplasmic reticulum are abundant or extensive in those cells which are synthesizing steroids. And RER are abundant in those cells which are secreting proteins or glycoproteins. Right. So guys, what do you think are these structures? These are mitochondria, obviously. They look like mitochondria. They have sausage shape, or we can say oval shape as well, right? These are mitochondria, and this is the nucleus. Right. Guys, this is the electron micrograph of a phagocyte engulfing bacteria. So we can mention over here is that this is basically the electron micrograph of what? Of a phagocyte, which is engulfing bacteria. Engulfing bacteria. So over here you can see that what are these? These are engulfed bacteria. And obviously bacteria are found inside what? Bacteria are found inside the phagosome. So guys, this vacuole that you see is the phagosome or the phagocytic vacuole containing a lot of bacteria, right? So this is something which is different that can arise in the paper. Over here we have the nucleus of the phagocyte, right? And what are these structures? This one, these are mitochondria. Where are exactly the lysosomes? I will let you know. So guys, in this electron micrograph, lysosomes are darker, right? And where are the lysosomes? These small structures over here, these are lysosomes. They appear darker. So how do we distinguish lysosomes from the mitochondria? Mitochondria will be larger and lysosomes will be smaller. So all the mitochondria will roughly be of same size, right? If you see a dark structure of smaller size, for example, so these will be lysosomes, right? So what are lysosomes? In this diagram, these are the lysosomes, right? Which will fuse with the phagocytic vacuole to destroy the bacteria. Guys, this is another cell. This is another animal cell. This is electron micrograph of which type of animal cell? Chondrocyte, which is a cell that makes up the cartilage. So we can see that in the center we have a large nucleus. And what else can we see? We can see RER. So these are RER. How did I recognize that this is RER? Again, you can see dots above the sacs. What else can we see? We can see these particles which are sausage-like and spherical. These are mitochondria, right? Okay, this is another electron micrograph taken from an animal cell. So what do you think is A? A is the nuclear envelope, right? And what do you think is B? B is the mitochondrion, right? What about these structures? I told you that if you see two cylindrical structures at right angles, these are centrioles which are making up the pair. What do you think is C? C is the lysosome. And can you see these curved structures? These are Golgi bodies or Golgi apparatus, and these are Golgi vesicles, right? So we can label these as Golgi apparatus, and coming out of the Golgi apparatus are what? Golgi vesicles. Right. So now we can move on to the diagrams of, or we can say to the electron micrographs, not the diagrams, the pictures, or we can say the electron micrographs of individual cell structure. So guys, this is an electron micrograph of a nucleus. And how do you identify the nucleus? It will have some dark areas and some light areas. Dark areas are what? The heterochromatin, and the light areas are euchromatin. And you can also find in the section a large dark area in the center, which will be nucleolus. So guys, over here in the center we have a nucleolus. Nucleolus will be kind of spherical and large, right? This is the mitochondrion. We can see its characteristic sausage shape, and we can see the cristae, the lines in the mitochondrion, right? All right, this is RER, rough endoplasmic reticulum. And how do we recognize this? We will see sacs or pouches, and there will be dots on the external surface of these pouches. What is this, guys? This is Golgi apparatus. You can see curved sacs. This is a very good picture. Normally we are not able to see such picture of the Golgi apparatus. And these are Golgi vesicles. So this is a highly magnified image, and which is a very clear image showing the Golgi bodies, which are curved sacs, right? This is the image of centrioles, which are cylindrical structures at right angles or at 90 degrees to each other, right? These are centrioles. And guys, this is the cell surface membrane or the plasma membrane. So how did I recognize this? Because under the electron microscope, the cell surface membrane appears trilaminar. There are three layers. There are two dark lines or two dark layers, and there is a pale layer in the center, right? So basically this is the cell surface membrane. And why are there three lines, or we can say three layers, and why are there two dark lines and a central pale line? So basically two dark lines are due to hydrophilic heads, right? Two dark lines are due to hydrophilic heads, and the central pale line is due to hydrophobic tails in the membrane. You know the membrane structure. The membrane consists of what? Hydrophilic heads and hydrophobic tails. Membrane is phospholipid bilayer. So in the center we have hydrophobic tails, which gives a pale line, and at the edges we have hydrophilic heads, which give dark lines. So guys, we are done with the electron micrographs of animals.